Okay. So as you can see, the topic for today is forensic toxicology, uncovering truth through poison analysis. Now, what do we understand by toxicology? So toxicology can be known as a fundamental science of poisons. Very interesting as such. So it is a discipline of science which deals with the poisonous substances, which when enters the body of an organism causes disturbance in their normal functioning and lead to the harmful effect, the most severe of which may be the death of an organism. But then, do poisons always cause death to a person? We'll get to that later. So if we look into where toxicology exactly came from, it comes from a Greek word that is toxicon or also toxicum, which means poison, and logos, which means study. So if we define toxicology again, it involves the understanding of chemical reaction and interactions and biological mechanism. So we are very aware that there are different mechanisms that goes inside our body. And what does this xenobiotics or the poison that enters from outside to our body do? They alter these biological mechanisms and then these leads to the adverse effect and the most severe of which can be the death of a person. Moving forward, now what is forensic toxicology? The term forensic toxicology, it covers any application of the science and study of the poison to the elucidation of questions that occur in judicial proceeding. Now, with the judicial proceeding, we can understand that it is basically concerned with the medico-legal aspect of the poison analysis. Now, what do we understand by the medico and legal? So, medico is basically identifying what exactly lead to the poisoning of the person, and legal is for the judicial process and for to identify who the exact culprit is, who exactly poisoned the person. So we can say forensic toxicology is a highly specialized area of forensic science which requires expertise in analytical chemistry, pharmacology, biochemistry, and forensic investigation. Why does forensic toxicology has a lot of subjects also involved in it is because without all of these subjects coming into play, the poison analysis will not be successful and the toxicologist for that sake cannot be coming to any conclusion. So he or she requires an expertise to have an expertise in the subject of chemistry to have a better understanding what exactly is the chemical structure of that particular substance or that particular poison, to understand how does the poison enter inside the body, how it exactly gets absorbed, distributed, then metabolized and excreted for that say, the biochemistry of it, and then the forensic investigation which will help to identify who exactly is the culprit or the perpetrator in that case. Moving forward, now what do we understand by poison? So, poison is any substance, irrespective of its quantity or quality, when given with intention to endanger, injure, or kill a person, is called a poison. Now, before we move further to the poison, I would like to put a quote here given by a very famous scientist in the field of toxicology, that is Paracelsus. He says, all of the substance are poison. That is the dose which differentiates whether a substance is going to act as a poison or it will act as a remedy. There is a thin line difference between whether a substance will be acting as a poison or it will be acting as a remedy for another person. Now, what is that thin line difference? That thin line difference, it comes from the dose, that what amount of dose has been administered to the person, that will be the one which will be defining whether that substance will be acting as a poison or it will be acting as a remedy. Let's take an example here. We are very aware of a tablet that is paracetamol, which a person takes usually if he or she is suffering from fever, for that sake. But then, let's say 250 mg of paracetamol or 500 mg of paracetamol is acting as a remedy or as a medication to a person. But then, if some person is, is undergoing some depression or some anxiety and has suicidal intention, the person goes and take a lot of paracetamol. Let's say he takes some 1000 mg of paracetamol, that makes some two tablets, or let's say he's taking some 3 or 4 tablets altogether. This leads to acting as a poison for that particular person and which can lead to the death of the person. We can also define poison as such. It is a substance which can be, it can be solid, it can be liquid, or it can be gaseous, which if introduced in the living body or brought into contact with any part thereof, it will produce health or death by its constitutional or local effect or both. Thus, almost anything is a poison as mentioned by Paracelsus as well. So now, if we look into poison, it can be endogenous or it can be exogenous. Now, what do we understand by endogenous poison? poison which is formed within the organism itself. Let's say some person, we have lot of enzymes, lot of different things which are present inside a person's body. But then if we, our body give us sign and symptoms if we are having some or the other disease as such. Let's say you are having vomiting, you are getting fever from some continuous 1 week or 4 5 days, and you're not able to identify what exactly is happening to you. You obviously go to a doctor and try identifying what it is. And then when the blood test is done, you will be given a report which says you are having so and so level which is high in your body. That itself is acting as an endogenous poison for your body. Now coming to the next, that is an exogenous poison, that is enter the organism from the outside, which is also known as xenobiotics. So what exactly is a xenobiotic? Any poison, any chemical or any pollutant for that say which is present in or around us can act as a poison, can act as an exogenous poison for a person. Now, if we look to the broader classification of poison, there is no clear-cut distinction for how we can classify poison. Depending on what a person wanna classify it as, the poisons are classified. Here, the classification that I have presented is based on the mode of action of a poison. So, if we look into it, the basic three major classification here are: first is corrosive, then we have irritant, and then we have systemic poison. Now, what do we understand by corrosive poison? As the name itself says, corrosive, that means whenever this poison come in contact with a person, it leads to the necrosis, that is the death of that particular tissue or area, whichever it comes in contact with. And then again, it gets classified as basically strong acids and strong base. So we know strong acid or strong base, whenever they come in contact with the body, what they do? They lead to the damage or destruction of that particular area wherever they come in contact with. We all have heard a lot of acid attack cases going on, and there we use the what acid is used there is known as sulfuric acid, and that sulfuric acid is used to basically
Um, Hello, Richa. Please unmute yourself.
Am I audible now?
Yes, now audible, and the slides are not visible. Please check that.
Oh, okay, sure. I'll check that again.
I think we lost your network connection.
I'll just check, Mamta. Thank you for telling. Are my slides visible now?
Yes, now visible.
So I'll go through the classification of. Can you tell me where exactly did I got cut off? Or I'll do the classification of poisons again. That's not a problem.
Yeah. Start from here.
Yeah, okay. So as I was saying, there are different classification for the poison. Here the classification that I have presented is based on the mode of action, how exactly the poison affects when it enters inside the body. So the first classification in this category is corrosives. As the name itself says corrosive, that is whenever it comes in contact with a part of the body, it leads to the corrosion or the necrosis of that part. In that we have two broad categories, that is strong acid and strong base. Now, we are very aware of different acid attack cases in which people use sulfuric acid, or which is also known as oil of vitriol. We also have other acids in that category, that is hydrochloric acid, nitric acid, acetic acid, hydrocyanic acid, etc. Now, where can we find this hydrocyanic acid in our daily life? We actually get this hydrocyanic acid from the apple seeds as such. Then in strong base, we have sodium hydroxide, sodium carbonates, and etc. Now moving to the next category, that is irritant, we have inorganic, organic, and mechanical. In inorganic category, we have metallics. Basically, all the heavy metals fall under this category. And in non-metals, we have chlorides, cyanides, phosphates, sulfates, etc. Then moving further, that is organic, which is basically classified as animal and vegetable, or you can say plant poisons. So in animals, we have snake venom, we have scorpion venom, we have spider venoms, and then in vegetables, we have Canade, Datura, Atropa belladonna for that say, Colocasia, etc. In mechanical, we have diamond dust, dry sponge, glass dust, etc. From where exactly do we get this mechanical poison? Is from wherever a person is working, let's say in some cement industry or some silica industry, there they have a lot of exposure of these all dry sponge and glass dust for that sake. Now moving to the further, that is systemic poison. Now systemic poison has a more broader category. Now systemic poison, what do we understand by systemic poison? Systemic poison is nothing but what exactly which poison affects to which part of your body. Depending on that, the classification has been made. So cardiac, it basically stands for the cardiac poisons. Basically, it affects the cardiac or the heart. That is the very common example for this is digitalis, nicotine, or wolf's pain. Then spinal, which basically affect the spinal cord, and that is nux vomica or gelsemium. Then we have cerebral, the broader classification we'll discuss next. Then asphyxiants, basically these causes failure of the respiratory system because it leads to the oxygen deprivation, carbon dioxide, war gases, tear gases, mustard gas for that say, and peripheral, which affects basically the peripheral nerves of our body, basically curate and conium. Let's move to the cerebral poisons. The cerebral poison has a very broader classification. The first one in that category is CNS stimulant. Now, what do we understand by CNS stimulant? It is the drugs which basically stimulate or gives an up effect to our CNS or the central nervous system, like cocaine, nicotine, imipramine, etc. Then CNS depressant, which basically gives a down effect or basically depresses our central nervous system, like barbiturates, tranquilizers, alcohol, anesthetics, opiates, etc. Now, deliriums basically alters the perception of a person, but then they are more active or they're longer acting as compared to hallucinogens. Deliriums and hallucinogens are somewhat related, but the difference being deliriums, they alter the control of a person. The person is not able to understand. But then in hallucination, it is more like the person can differentiate what is real, what is not real, but then not to a greater extent. And deliriums have a more acting period as compared to a hallucinogen drug. Now, moving forward, we'll see what are the characteristics of an ideal poison. Like, how does a person decide what poison should be suitable in case he has a suicidal attempt or he has a homicidal intent in his mind. So, if we consider accessibility, that is, in suicidal poisons, they are very easy and free. They do not bother much whether it's gonna be how is it available or that way. But then in homicidal, it is more like they make sure it is resembling to some or the other thing so that the poison cannot be detected as such. If we consider the lethal dose with the suicidal poisons, it's like a very small dose should be lethal enough. But then homicidal also the very same. The very small dose should be lethal because if it has a large dose that makes it lethal, then it will get detected in longer run, and then in toxicology as such, it will be eh, getting in the reports, which is not advisable if a person is killing a person. But then next we have lethal period. So in suicidal, again, it is small, and homicidal, it is long acting. Why homicidal poisons are expected to be long acting? Because if let's say a person is giving certain poison to a person, and then he wanna escape from there. If it is a very small acting poison, then he won't have a time to run away because he himself become a perpetrator and he will be caught then and there. But if the poison is long acting, it will be very difficult for the sign and symptom to appear, and then someone can identify, and then it will be a longer process, and then the person can escape. Taste wise, it is like pleasant taste for both the cases. Sign and symptoms in suicidal, as such, we do not consider much. But then in homicidal, the person has this in mind when they choose the poison as such, it should resemble some or the other disease so that they cannot identify what exact poison was it that was given to the person. Now, some very common examples which usually are used as a suicidal poison, insulin, because it's very accessible. Usually, people who are suffering from diabetes have the access to it, and then people who have the suicidal intent can take just this insulin injection, inject them, and they can have this. Then cyanide again with apple seeds themselves, but highly not suggested. Opium again. And then in homicidal poison, we have organophosphorus, that are different type of pesticides and agricultural fertilizers, which are very available basically in village areas, or let's say people who are into farming and stuff. So it's very common, and it's a very common type of homicidal poison which has been encountered. Then we have strychnine, arsenic, aconite, barbiturates, thallium. Now, if we see different types of heavy metals are the one which are basically used for homicidal poisons. Why? Because they have this very small fatal dose, but then they have this very long acting time. With a very small dose that you keep on giving to a person, it can lead to the acute poisoning of that, chronic poisoning of that person. And by the time the person can themselves identify, or by the time it affects the person, it has been quite a lot. Let's say 1 month or 3 months have been passed for that say, and then it finally comes to notice that so and so poison was given to the person and he has passed away because of it. Now, moving further, what effect does the poison exactly cause to the body? So basically here we have three things, that is local, remote, and systemic. So local effect of poison, or you can say local action of a poison, what do we understand by that? Effect can be seen at the site of application. That means wherever the poison come in contact, the effect will be shown then and there itself. Let's say the poison has been applied to a person's skin. The effect will be shown in the skin itself, at the site of application. If the person has ingested the poison, the inflammation will be seen in the mucosal membrane for that sake. For these, we have very common example phenol, that is also known as carbolic acid, and then irritants such as which leads to acid inflammation. Then remote effect of poison is distributed from point of entry. That means first the poison is ingested, then it gets distributed to different part of the body, and then it shows its action for that sake. So it does not have any local action, but first it gets absorbed and then it shows its action. In case of atropine, that it leads to eyes and mydriasis. And then the systemic, which is actually a combination of both local and remote effect. That means the poison will show its effect in the local, that is the site of application also. Along with that, it will also show its effect after getting absorbed further into the body. The very common example of this is oxalic acid for that say. Oxalic acid, when ingested in larger quantities, it leads to the formation of calcium oxalate, which is very synonymous, or also we can say kidney stones is also one of the common example of this. Highly not suggested as it can lead to kidney stones. Moving further, what are the factors which actually affect your poisoning? So we have two factors again here. One, the poison related, and the other one that is patient related. First, we'll see what are the poison related factors which affect poisoning. So what is the concentration, or let's say what is the dose? Now, the higher the concentration, the greater the toxic effect or the adverse effect. If a person is given a very small dose of a poison but for a longer run, then it leads to the chronic poisoning of that person. But then, if the very same poison is given to the person in higher dose or in higher concentration, it leads to the acute effect or acute poisoning of that person. Now, solubility. The higher the lipophilicity, the higher the toxication. As our body has this lipophilic region, so what happens when the drug or the poison is lipophilic, it gets soluble in it, and because of it, it leads to the more toxication of the person. But if a drug or a poison is hydrophilic, the toxication or the level gets decreased. Then route of administration. By what route the drug was given to the person? So, if the drug was given to the person or the poison was given to the person with intravenous route of administration, as it is 100% bioavailability, because it directly goes to our systemic circulation. There is no scope for the drug to get absorbed because it directly goes to the circulation and it can directly get distributed in the body for that sake. And that is how it acts fastest in the body. And if the person gives any poison with the help of intravenous route, it acts the fastest. But let's say if the person ingesting a poison, or let's say he has been given the poison with the topical route of administration or intramuscular or any other route of administration, the time of acting varies and the poison for that say will have different effect because it will have less bioavailability. More time will be taken for it to get distributed, then get metabolized, and finally show its action for that say. Now, if we come to the patient related factors, the first factors is the age. So, if pediatrics and geriatrics, so pediatrics are basically the children, basically, and those children, as they are not fully developed, and then what happens if we are giving some drug to them in a larger dose, the drug start getting accumulated, even if you give them in that normal dose itself, because the children they have this phase one metabolism developed in a but then their phase 2 metabolism is not that developed, and that is the reason they're not able to completely metabolize the drug or the poison given to them for that say, and it leads to the accumulation even in the normal dose. But then if we consider the geriatrics or the people senior citizen, basically who are above 20 to 40 age circle, so what happens for them, their metabolism is very slow, and that is the reason they are not able to metabolize the poison, and that then it affects them more. If we consider habits, let's say a person is smoking cigarette. Now, what happens exactly if a person is cigarette smoker? So it leads to the enzyme induction. And if a person is alcohol consumer, so alcohol again it can have both acute and chronic effect to a person. If we talk about the acute effect, it can act as an enzyme inhibiting. But then if we talk about the chronic effect, it again leads to enzyme induction. Then tolerance. So, what exactly tolerance is? If you yourself see while you start consuming a certain medication, again coming to a paracetamol example, if you're continuously taking paracetamol of 500 mg every time you're getting fever, but then after some 10-15 times, you yourself can see that 500 mg is not effective to you anymore. Why? Because your body has gained tolerance towards that 500 mg of paracetamol, and the fever is not going. And then you'll have to increase the dose as such to reach that particular level of therapeutic dose for your body as such. So that is how the tolerance work for a person. So if a person is again a alcohol or drinker, so what happens for them also is like they'll keep on consuming the alcohol for a longer period of time, and then they start gaining tolerance as such, and then they'll have to consume a whole lot of alcohol to get the very normal level of high or the kick that they want for them. The very same way in the poison also, if the person is consuming continuously, let's say opium products or any cannabis product for that say, if they continuously keep on taking it in smaller dose, then their body becomes tolerant to those poison, and then when they need a higher kick also, they'll have to increase the dose of those things. Now, idiosyncrasy. Now, what exactly is idiosyncrasy? So, it is basically genetically determined. It is a form of allergic reaction which is not very common to all the patients, or all the person, but a few of the person. Let's say some of the person of having this allergic reaction if they eat peanuts, or some of them are having allergic reaction towards mushroom, but then other people are not very allergic towards these peanuts or these mushrooms. So it is just based on their body functioning or their genetical makeup that they are very allergic towards such things, which is common only to a fraction of people and not relevant for other people as such. Now, moving further, coming to the forensic analysis part, that is, what exactly a toxicologist as such is supposed to do. Like, first, he'll start with the evidence collection, which basically is done by a forensic expert, then evidence analysis, and then evidence presentation. Now, what are the different evidences that we can collect from a crime scene? First, if we start from the crime scene, let's say there has been a case of poisoning which has been registered as such, and then you as a forensic expert has been called up to collect the samples from the crime scene. So what are the different samples that you think you'll be collecting? So first thing that you'll be collecting, if you're suspecting that there has been some malicious intent or some poisoning has happened there, so you'll be collecting whatever food you are getting, whatever let's say and if there is any alcohol bottle, or you're seeing any cups or glass or anything which has some or other liquid present in it, you'll be collecting all of it. But as a control sample also, you will be collecting whatever is present in the larger vessel in the kitchen as such, to identify whether the poison was mixed in the complete food that has been made, or whether it was just present in the food that the person has consumed. It was mixed there or it was mixed from the start. Now, what else can you collect is, in case if there is any medication which you are suspecting that the person has taken a whole lot of pills, because usually we encounter a lot of people consuming overdosing on different medication like sleeping pills and stuff. So if there is any medication as such which you see a lot of tablet wrapper are there which has been popped open, and then you're gonna collect all those tablets for that sake. Now, we will move further. What are the different evidences we will collect as such? Let's say what happens next is, first you as a forensic expert, you go to the crime scene, you collect these samples. Now the body is sent to the mortuary. Now what happens there is, the doctor, basically the forensic medicine expert, he conducts the autopsy, cut opens the body, and then collect the visceral sample. Now what exactly are the visceral sample? Basically all the internal organs along with the different body fluids that is present in our body are considered as visceral organs or visceral fluids for that sake. Now, why exactly do we have to collect these visceral samples for the analysis? Because these samples are the one which will help us to identify what poison exactly was given to the person. Now, what are the different visceral fluids that you will be collecting? So the first visceral fluid that is usually collected is blood. Now, why do we collect blood and where do we collect it from? How exactly we collect it? How much quantity of it we collect it? What preservative we collect it? All of these are very important, because let's say you have collected some 10 ml of sample, but then if the test that the toxicologists do is not, it goes wrong or you get false positive results, so you'll have to have more samples so that you can run the test all over again, excluding the factors which led to the false positive result and getting better results or a accurate result for that sake. So first the sample, that is blood. So blood, basically it is obtained from the femoral artery or vein by percutaneous puncture and using 50 ml syringe with a wide bore needle. We try to collect approximately 50 ml of blood, but at least 10 ml should be there. And then how exactly the analysis proceed further is, when these samples are taken to the forensic toxicologist, they divide the sample into three parts and then only they start their analysis. Why do they have to divide the samples into three parts? They divide the samples through three parts, basically control and then first and second. We are always supposed to have a control sample to make sure that the result that we're getting is correct or not. And then we have two more samples, the one which we will be using for the analysis, and the second one we are having is to check whether, in case if the first sample gives you a false positive result, you're having a separate base of sample which you can use for the further analysis or rectifying your mistake for that. In case you're collecting blood, you will use sodium chloride or potassium oxalate as your preservative, and then you use EDTA and heparin for anticoagulation. You always pack your blood sample in vacuum containers and then you send it to the lab for the analysis. Moving next, we have urine as a very important sample. So urine in dead, it may be collected by direct puncture with needle and syringe of the exposed bladder after the abdomen has been opened. Or it may also be obtained by insertion of the urethral catheter before starting the post-mortem. We basically use sodium fluoride as preservative, but then there is one more preservative which we can use in context of urine, which is also known as thymol. Then, as we can see, the most or the let's say the major sample that we usually get or always get in context if there is a suspected poisoning, we always get urine and we always get blood. Now, why do we always need to have urine as our sample? Because as we know, liver is the major organ where exactly the metabolism of the any poison or anything, any food or any drug takes place. Now, after it gets metabolized, it gets excreted with the help of kidney does that functioning. So after the kidney has done its excretion functioning, what happens? It goes out through urine. So whatever metabolite of that particular poison or that particular drug is present will be very actively present in your urine sample, and that is the reason urine is one of the best or one of the major sample that we usually collect in suspected cases of poisoning. Now, moving next, we have bile. Bile, as such, is not collected in all the poisoning cases, but in certain cases, if there is suspected case of cannabinoids or opioids, we usually collect bile. It is aspirated by a needle after the abdomen is opened and before the organs are removed. All available bile should be removed from the gallbladder and preserved with sodium fluoride. So bile as such is not collected, but then again, yes, it is very helpful for the estimation of different poison, basically the plant poisons, and it can be identified with the help of your bile. And then they are again preserved in different amber colored bottle. Now again, why also do we use amber colored bottle for the preservation or the of your different visceral sample? We use amber colored bottle because let's say you have packed a sample and then it is while transportation it is getting exposed to sunlight. Now if it is amber colored, what happens is it stops the photo oxidation which can be happening because of the sunlight being there. So that is the reason we usually prefer amber colored bottle for the packaging of our different visceral fluids. Now, if we move next, we have cerebrospinal fluid. Cerebrospinal fluid is also not collected in every cases. Mostly it is collected only in the cases when we are suspecting that some cerebral poison has been ingested or was given to the person. So in this case, we usually make a lumbar puncture or we aspirate with the pasture pipette from the base after frontal lobes of skull has been opened, and the maximum possible amount should be withdrawn. We usually use sodium fluoride as a preservative in this case also. We can also collect it from the atlanto-occipital region of the skull. Now, moving next, that is vitreous humor. We usually collect 2-3 ml of vitreous humor from one or both the eyes, is aspirated from the lateral angle of the eye with it a 5 ml syringe. Now, this vitreous humor is the last thing which did I lose my connection? Hello.
Please, you're audible. And
Am
your
I
slide
audible?
is visible. Yeah.
Okay. Thank you. So vitreous humor, it is the last thing which gets decomposed for that say, and then if the stages of decomposition is started in a body, after some 3-4 days, this vitreous humor usually turns brown and cloudy. And then there is this potassium level which increases further, which will be helpful to identify what time has been passed since the person is dead. Now, if we consider there are some other, that is one more visceral fluid that we can collect, that is known as synovial fluid for that say, which is basically present in our knee joints, and then it is also helpful for the analysis of what poison is present basically in alcohol cases. Then next, what are the different visceral organs that we collect? So first is your stomach and gastric content, because let's say the poison was ingested via food or any drinkable item, so we collect all of it. And then if there was some pill which is present in very intact form, we collect it in a different pill box. Then kidney, we collect minimum 25 gram of the kidney. And then liver, because liver is the major site where the metabolism happens, so we collect liver, and we do not collect the liver specimens from the right lobe are preferred. Why? Because the left lobe is connected to it's more towards our stomach, so there is a possibility that the diffusion can happen and it can lead to the contamination. So that is the reason we do not collect from that part. And then lungs basically if there is any asphyxiants or let's say any poisonous gas which is involved, so we collect lungs. Now for lungs we can also use glass jars for the preservation or packaging, but then we have this nylon bags which are available that we mostly use for the collection of lungs. Now why do we prefer nylon bags? Because nylon bags it traps the air or the poisonous gas which you exactly are supposed to do the analysis of to identify how exactly it led to the death of a person with the asphyxiation. Apart from this, we also collect spleen, we also collect brain, but then we also collect heart. But then brain and heart are not that advisable until and unless it's suspected. Like for brain, it's like some cerebral poison is involved. For heart, it's like some cardiac poison is involved. Then only we do the collection. But majorly, it's your stomach and gastric content and your liver and kidney. We also collect bones and hair and nails in cases we are suspecting some heavy metal poisoning has been involved. Now, how exactly we do the packaging of all these evidences? So basically, we use wide mouth glass bottles, which has a capacity of some 1 to 2 liters for the collection. And then we seal it properly, write all the details, whatever has been asked, like the FIR number, the case number, the name, the age, the gender, all the basic details should be present there. We seal it properly and then we send the sample for the analysis to the forensic science laboratory. Now, once the sample are ready to be sent to the laboratory, there are different documents also which are supposed to be sent along with it. Now, what are the different documents? So first, there should be a copy of Panchnama and FIR. If it is available, brief facts of the case submitted by police and case sheet. Now, what all things should be again present? It should have all the details, a brief summary, what exactly is the case about, what is the name, who are the relatives, what exactly happened, if there is any medication, whatever details can be gathered is put together in it and it is sent for the to the forensic science laboratory for the person to or the toxicologist present there to understand what exactly the case is all about. Then a copy of the autopsy report with detailed observation and finding. Also in this, the poison which is suspected, because as the doctor do the autopsy of the person, when he cut opens the body shows different internal sign and symptoms which help or which gives a guess estimation what exactly can be the poison that was given to the person which led to the death. So that poison can also be mentioned so that a whole lot of criteria, a whole lot of poison can be excluded and only some possibility or some category of poison can be used for the analysis, because we have very limited sample that is sent for analysis, and if we just keep on doing analysis or test for all the samples for that, say, we will not be reaching to any conclusion. Along with this, we send a forwarding letter, which is by the investigating officer requesting to examine the submitted sample and informing the finding to the medical officer and indicating the viscera box is transported by the constable who is authorized to do so. And then authority letter from a magistrate or relevant officer for chemical examiner to examine the visceral sample. Without any of these let's say if the sample has been sent, but then if there is the forwarding letter or the authority letter or any other thing is missing, any other document is missing, the FSL or the forensic science laboratory people, they will not accept the sample, because once they accept the sample, it is completely their responsibility. So they do cross check whatever requirements they're having is all met, all the details are completely filled, everything is intact, and then only they take the sample in their responsibility, and then they proceed further with the analysis. In case anything is missing, they'll just return the sample back until and unless it is again clarified, sent back, then only they'll accept it. Now, coming to the final part, that is sample analysis. Now, how exactly the sample analysis work? So, you did the sample collection. Once the sample Now the sample has been sent to the lab. Now you as a forensic toxicologist is asked to do the sample analysis for the different visceral sample that you're receiving. Now, how will you do the sample analysis? So once you receive the sample, you just check what is the case history of it to you get a better understanding of what exactly is the case all about, what sort of poisoning can it be, because based on the different sign and symptom which is mentioned in the autopsy report, you can identify yes, so and so can be the poison which is present there. So first you receive the sample. Now your main aim is to extract the poison out of that particular sample. You cannot just take a sample and start put it in your GCMS or let's say any other instrument, HPLC or for that sake. You first will have to take the sample and then you extract the poison out of it. Now, how exactly do you extract the poison out of a sample? There are different extraction method that we have. The very common being liquid extraction or solvent extraction, in which we use two immiscible liquids, and then these are basically best for biological matrices for that say. Then we have solid phase extraction or solid phase micro extraction. We have distillation or steam distillation. We have fractional distillation, which is best used for if let's say kerosene is mixed with some other type of oil or in that case. So once you do the sample, once you isolate or you extract the poison out of the sample, now it gets divided into further categories. Acidic, it can be an acidic drug or it can be a basic drug. But then it's not mandate that every time it gets divided to either an acidic category or a basic category. Depending on what classification you are using as such, it can get categorized. Here I am using as acidic drugs and basic drugs. Now the next thing is once I have extracted the poison out of it, now I do the screening test. Now what do we understand by the screening test? Screening test can also be known as a presumptive analysis or presumptive test or a preliminary test. Now what exactly happens in this preliminary test is we do different color tests or we use thin layer chromatography to identify what type of poison is present. Let's say I'm suspecting it is a CNS depressant that is present over there, or let's say I'm saying it is some plant alkaloid. But then we have plant alkaloids which fall both in acidic category also and then in basic category also. Now, how exactly will we know? We have different type of color test. We have Duckness Levine test. We have Banner reagent test. We have a whole lot of tests for different type of plant alkaloids, which can help us to identify depending on what color they are giving, what is the poison. That excludes a whole lot of other possibilities and just leaving me with one type of possibility. And that is how I proceed further. Then we get this curve. We move further once we have an idea. Let's say the cannabis is present after the screening test. You identified you are getting this color, and then you say the cannabis is present in the sample. You go for the confirmatory test. That is, now you are using different instrumentation facility that is available in the toxicology lab to get a clear answer whether the cannabis exactly is present or you are getting a false positive result for that sake. So we have different confirmation tests or we have different instrumentation that we can use, like we have GCMS, that is gas chromatography and mass spectrometry. We have high pressure liquid chromatography. We have atomic absorption spectroscopy. We have ICP-MS, that is inductive coupled plasma mass spectrometry, which can be used for the analysis of it. Now, basically, we can use GCMS and HPLC for your plant alkaloids. You can use AAS and ICP-MS for your heavy metal analysis. You cannot do your heavy metal analysis in your GCMS or HPLC for that sake, right? Now, moving further, what are some of the common poisons which we found in Africa? So we find this elephant's ear, which is also known as colocasia. We have arum lily, and then we have Nereum oleander. So if we talk about the mechanism of action of colocasia and arum lily, they both have this oxalic acid type behavior, meaning they have this calcium oxalate which is present in their leaves as such, which actually has a good effect in the body, which the people there in Africa thinks it's good for them. But then if they keep on eating it on continuous basis, it actually releases this calcium oxalate needles, which pierce the tissues inside the body, goes inside and leads to the kidney stone, which is again very poisonous for the body. And then it also shows oxalic acid type poisoning. and that is how it leads to the poisoning of a person. Now, if we talk about medium oleander, it is a very good cardiac poison that is available, and that is how it basically leads to cardiac arrest and is very toxic to the person. Now, moving next, what is the role of a forensic toxicologist? Let's say you are going to some FSL, that is forensic science laboratory, and you're getting appointed as a forensic toxicologist. So what will you be doing? So first thing, you will evaluate determinants and contributing factor in the cause and manner of death. That is to identify what exactly is the poison which led to the death of a person. That you will be doing by different, that we just discussed the sample analysis part. From there, that is what you will be doing. You'll do the extraction of the visceral sample, then you'll do the screening test or the presumptive test, and then you do the confirmatory test, and then you will be giving a report ki so and so poison is present in the visceral sample that you have been given with. Next, collecting and testing for harmful contaminants in food or the environment. It's not just sometimes you also get samples. Let's say you have seized, or there was this truck which was going carrying illegal substances, so you also get those cases. So it's always just related to the dead body. Sometimes it's about some drug or something which has been confiscated from some rave party or some truck which is carrying illegal substances. And then you use different chemical and biochemical instrumentation, and then you do the identification, and then you do provide the expert testimony, because whatever result you are producing will be the one which will be presented in the court of law as an evidence, and then you will be called upon to give the testimony to justify your result if the court is not satisfied, if the opponent lawyer is not satisfied, whatever their doubt is. Now, again, one more thing to keep in mind, that our main aim as forensic toxicologists is to identify what poison is present in the person's visceral sample. But then the qualitative analysis is to be done. Quantitative analysis comes next, and it is done when asked as such. Or if you are doing some research, if you encounter some poison which is very new, there has been no studies as such, then you move to the quantitative analysis to identify what quantity can be fatal, or what is its fatal period, studying the complete chemistry or the complete biochemistry of that particular poison for that sake. Now, moving further, I like to discuss about a case study. This is about thallium. So thallium is basically a metallic poison which is actually a heavy metal and which is coming to limelight a lot recently. It is also known as a poisoner's poison, or you can say inheritance powder. Now, what exactly this thallium is doing is, so what happened? First, we'll discuss what happened in this case study. So, in this case study, it is of New Delhi, India. So, there was this person, there was a couple. They had two children already, but then the lady conceived the baby. But because she was having complications, she wanted to abort the baby, and her husband was not ready to get the baby aborted. Why? Because the husband's father died the very same year, and then later on, the lady conceived the baby. The husband thought the baby is gonna be the reincarnation of his father. Now, as the lady was having the pregnancy complications, she, along with the lady's parents, they decided they will get the baby aborted, and that is what they did, going against the husband's will. Now, this husband had to take the revenge, so he poisoned the complete family of the lady with thallium. He mixed thallium in the food, basically the fish curry that he prepared. He called them for the dinner, basically the lady's father, mother, sister, even the maid. He poisoned all of them. So the mother and the lady's sister, they ate the curry. They were admitted to the hospital. As thallium is a slow acting poison, its severity is visible only after good 2 to 3 weeks. So once the poison was given to them, they had this breathing issues, they had high heart rate, high blood pressure, they were admitted. And later, when when after 2-3 weeks, even after the treatment, uh, they were not getting rectified as such. And then the doctors informed that both of them have passed. Then the father and the maid, they were still undergoing treatment, and the actual lady who was pregnant, that is Divya, she was in coma. And then what happened, now they, as they were suspecting, because when they did the blood analysis for them, they found out thallium in in their blood. Now they started linking all of it together, ki how is possible that everyone was poisoned with thallium, or how is it possible that they had even thallium in their vicinity. So when the police personnel started doing their investigation, so when they went and met the guy, so he told yes, he had given them thallium. They recovered thallium in a glass bottle from their home as well. And then he confessed that he himself has mixed the thallium in the food and given to the person so that he can take revenge, because the lady went against him and aborted the baby for that sake. So this is one example how exactly the thallium, which is a very silent weapon which kills person, and it is also a heavy metal. And there are a lot other heavy metals which exactly lead to the death of a person. Like we have arsenic, we have mercury, and there have been lot many different cases associated with them. But then thallium is the one which is coming to a lot of limelight these days. That is all. Thank you.
So please, if you could check the text box, we have some questions or some concerns there. Could you address them for us?
Yeah, yeah. Can you repeat why amber color bottle? Okay. There is this question, why do we use amber color bottle for the preservation of visceral samples? So basically, amber color bottles, they have this brown coloring. So this brown coloring, it protects the sunlight from entering, and then the photo oxidation cannot happen, and the sample which is present will not get oxidized further and leads to the destruction of the sample or the poison that is present in that visceral sample or visceral fluid as such that you are collecting. So that is the reason why we use amber color bottle, as it avoids the photo oxidation. Any details how this case was detected by forensic department? So what I found out was, it was basically the hospital people who collected the blood and then sent it to the lab for the analysis, where they found out that the thallium was present in the blood, and also the urine sample that was collected with the help of the different instrumentation. Mostly they might have used AAS or ICP-MS for the detection of it, and that is how they came to know that thallium is present, and that led to the poisoning of those person. And when the thallium was also discovered, or the bottle was recovered from their home as such, that was how it came into identification. Yeah.
Hope you don't mind sharing your email with them.
I can type in the mailbox only, the chat
Okay,
box, sorry.
so you kindly share your email. If anyone has any further questions after this lecture, they
Yeah,
can
sure.
reach out to you for more clarification.
Yes.
Also, I know definitely someone would ask, would you be sharing the presentation with them, or they should email you for that?
Sure. I can share the presentation with whosoever contacts me, or I can share it to you. You can share it with them if that is also okay. Anything is fine.
Okay, fine. All right. So those
Yeah.
who registered, I'll be sharing it with them if once I receive them from you. And also
Yeah,
anyone who has,
sure.
I don't have their email, can also reach out to you personally for it. Okay.
Yes, sure. No worries.
Yeah. So I would ask this question, then I can now open the floor to the audience if they also have any question. But
Yeah.
I wanted us to look at this scenario. It's something that I just thought about right now. Let's say we understand that all substances can be poisons depending on the dose, but let's talk about poison as in poison, like actual poison, something that we just know that is poison, not and also compare it to normal pharmaceutical drugs. Is okay, and look at them in terms of tolerance. Let's say
Yes.
antibiotics. It is being said that like the more you take antibiotics or the more we gain antibiotics into our system, either actively or
Yeah.
passively, with time we kind of build that how will I call it? We make and we kind of build that thing we call like antibiotic resistance in our systems, whereby in future if you get an infection, if you take the antibiotics, there's no more able to counter or give us the necessary therapeutic effect that we need. Now, also look at it in terms of tolerance, but now we are looking at, let's say, poisons like maybe, let's say, lead poisoning, like lead accumulation of poison. The more we get exposed
Yes.
to chemicals like lead, either
Yeah.
actively also or passively, yeah, with time, like our body doesn't gain that kind of like, how will I call it, level of tolerance, but rather it accumulates and it gives us, let's say, much more severe conditions like in the future. So why is
See, if
the
we
level of body? Yeah,
if we talk about heavy metals,
yeah, the
so
metals.
in
Yeah,
my knowledge, yeah, in my knowledge so far, whatever I have studied is like, or whatever I have came across, the heavy metal is something, which does not get eliminated from your body as such. It gets keep on accumulating. There is no as such proper metabolism happening for it or anything like that which can, eliminate it from your system. And then it leads to more and more accumulation, and then you cannot just, let's say, you cannot just randomly give a person 100 mg of arsenic or so and then kill him. Usually people do not prefer it. Why? Because if you're giving some person 100 milligram of mg at a time, the person will die very instantly, and then it will obviously be suspected that he was, poisoned for that sake. But then if you're giving it slowly, with that slow time period, it leads to the chronic poisoning as such, and it will become difficult to identify who exactly was the person who gave him, or was it, he was having him himself with some other thing or something like that. So the body does not get tolerant in context of heavy metals, because it completely alters the sulfhydryl group that is present in our body. But then if we come to pharmaceutical products, let's say I am taking paracetamol or antibiotics or anything. I'm suffering from certain disease and I started taking antibiotics or whatever. So now my body is getting recovered. But then if I acquire the very same disease again and the doctor give me the very same medication all over again, now the body will not react the very same way as it was reacting for the first time. Why? Because the person who has consumed it, the bacteria which is present as such, they get tolerant toward it. They develop this resistance, and then they can identify that the very same amount is given. Now they have this resistance and they'll counter it. So that is the reason sometimes the doctor also either they increase the dose as such of that particular medication, or they will change the antibiotic that can also be used. So as the antibiotics change, the composition will change, the mgs which is exactly present of it, that will change, and then the bacteria become again, the antibiotic become very new to the bacteria all over again, and that is how it works. So again, it doesn't lead to the poisoning as such. But then if the very same antibiotics is given for a longer run or kept on given continuously to the person, so what exactly is going to happen is the body becomes tolerant towards it, and then there is no effect or no use of taking that antibiotics as such for that sake. So that is how it actually works. If there is, I would like to look more on it. If there is any further clarification I can give for you in this context, I'll definitely look into it and I'll for sure mail this to you.
okay. The overview you gave is clear. I get that. I get that. Thank you. And also another question again. So looking at, let's say,
Yeah.
in terms of you are going to collect your samples, but the deceased or the body has been there for quite a very long time, and we know with that the body naturally clots the blood, and then you need some blood samples. How do you normally go by that? The situation whereby the
No,
body
no.
has been
Can
there
you
for
repeat
quite.
it again?
I'm saying you are
Can you
going
repeat
for
the
a
question?
sample. You
Yeah.
want to
Okay.
go for sample for toxicological analysis, but the
Okay.
body, the dead body has been there for quite a very long time, which means
Okay.
most of the blood or would have clotted. Like, so how would you go in that situation whereby you need blood to do your? sample analysis?
In those, okay. In those cases, mostly we try preferring blood, but then if it is already, being clotted, there is no point because we are not gonna anyways extract. We're not gonna get the exact state of it because let's say the body has reached this decomposition state as such, or let's say the decomposition is, getting started or anything like that. But then, if the blood cannot be collected, cannot be used because it has been clotted, we have these visceral organs, or let's say we have other visceral fluids. We can use urine, we can use CSF, we can use synovial fluid, depending on whatever is the case study, whatever the case study in the sense, whatever is the case history or the brief summary that has been sent as such, which will help to identify what type of poison it is. We get a brief idea from there itself. So depending on that, we just check, and then based on it, we collect whatever sample is needed in case we are not able to collect the blood. Mostly it is preferred if we get blood and if we get urine as such, but then if we're not able to get it, we go for the visceral organs for that sake.
Okay, well understood. So I'm opening it up to the audience. So Dr. Jayan, right, you raised your hand, please. You may have the floor.
Yes.
So, hello.
Hello, sir.
Yeah, I'm Dr. Jayan. I'm from AIMS Raipur. That is a national importance institute in India. So,
Yes,
as ma'am said correctly, we at times we do not get the poisoning, the traces of poison in the blood or even in the urine. So then we in India, because we have a very slow process of getting the reports, we usually give the visceral samples. Visceral samples are favored most of the time. Like if it is a lipid, if it's a it's a poison. Like for example, organophosphorus is very common in India.
yes, sir.
So we have this. It goes in the lipid stores. These poisons. So these lipid stores plus the viscera. If it is analyzed, we get the result. We get positive detection of this organophosphorus. So even though we do not get the same in the blood at times. So also hair and all can be also given if you are suspecting heavy metals. Hair can be also used. So that's it. I just wanted to add that. Thank you.
Yes, sir. Thank you, sir.
Thank you so much for the contribution. Please, any other suggestion, question, or contribution? I think Mr. Erasmus wants to say something.
Madam, could you type that investigations which you said in the box, please? Because I'm also learning these investigations, like which to do for which different type of poison. So kindly, could you for the knowledge of the audience, could you please share it in the chat box, which in poisoning, in heavy metals poisoning, which investigations would be preferable to be done, like for the detection?
So if for this particular case that I was discussing, that is thallium poisoning, if you want, I can share the complete case study as such from where I took what all findings I had. But then apart from this, if you want me like what heavy metal, what sample to be collected, how the investigation will proceed, I can make a complete different presentation on it and then I can share it. Will that be okay? Like it will be more informative that way, I guess.
Yeah, that's just like you can just repeat for now for time. I know that is depending on the organizer's availability. So if possible, could you just for the time being, could you just give me that names of those investigations? That one or two investigations which you said, like I think it is atomic absorption spectrum,
Yes.
which you are saying. Is it that?
So in case we want to do the confirmatory analysis for heavy metals, let's say, so we can use AAS, that is atomic absorption spectrometry, and then we can, or spectroscopy, and then ICP-MS, that is inductive coupled plasma mass spectroscopy. So these are like two. We have other variants also in this. We have ICP-OES, we have AES, we have AFS. So depending what is available in your lab, mostly it is like AAS and ICP-MS. These are very common, which are which are available as such, which are basically used for the heavy metal analysis.
Okay, ma'am. Thank you. Thank you so much.
Yes, sir. Can we consider a substance?
We have some question. Okay, you've seen
Yeah,
it,
I'm
right?
just checking that. We can also consider a substance a person can be considered a substance which make a person significantly impaired intellectual functioning as a poison, or a poison means it always leads. No, sir, the poison doesn't mean it will always lead to death. But then it is actually considered that it will lead to death for that say, because it actually alters the complete functioning of the body. Let's say there is some mechanism how exactly your body is working. But then if a poison is ingested to a person, it's supposed to be like it alters the functioning, it alters the mechanism, whatever biological interaction or reaction is going on, it completely alters it. But then, if there is something which has been ingested to a person which changes its or impairs its intellectual functioning, that is also considered as a poison, because the person was very healthy, but now something has been given to him which has altered its intellectual capability. Now he is not able to process things as he was, and this actually happens. Let's say when a person is taking certain hallucinogens, or there are certain medication for that. Say if a person is suffering from depression or anxiety, and then the medication sometimes they help them, but then apart from that they also have certain side effects at time which can also lead to their impaired intellectuality, which will also considered as a sort of poisoning that is happening to a person, because the person was very normal, he had this normal functioning, he was able to differentiate between things properly. But then now he randomly started thinking there is a let's say rope is present over there, and then he starts considering that rope as a what is it, that it's a snake instead of a rope. That basically happens when a person is hallucinating or having delusional thoughts. So all of these are considered as a type of poisoning that can happen to a person. I hope. Yeah, thank you. Yeah. Anything else? Is there anything else? Does the mere presence of the substance identified in the blood or body confirm poisoning? It's not just the mere presence of it, but let's say you were having some ailment and then you go to the doctor, you get your blood test done, and then there is something which is out of the blue which is present in your daily dose, in your daily report, you will be not getting those things as such. But then if you get some heavy metal present on it or something like that, obviously you will doubt, no, how exactly did it come to your body, or will you just sit and chill, no, right? So that is how exactly it happens. If there is some substance which is present which actually is not supposed to be there, so obviously you'll suspect some or the other type of poison has been given to the person, and that it will confirm that yeah, it is a case of poisoning. Alcohol can cause intellectual impairment, but usually only in severe doses it can lead to death. Yes, sir. You are correct.
Okay, Erasmus, I saw your hand. Please, you may ask your question. Mr. Erasmus, are you there? Please, any other question from the audience? Okay. Ma'am, any last or final words to our future upcoming forensic toxicologist?
I I just say,
Concluding
like,
remarks. Yeah.
you should be very aware of your surrounding first thing, but then apart from it, I'll also give a very basic example in this. All of us like having adventures in our life for that sake.
Hello?
But then, yeah,
Hello?
Erasmus.
Yeah, so am I audible now?
Yes, yes, you're audible.
Oh, okay. So this is Erasmus, Regent University, Ghana.
Yeah.
Yeah. So about 3 days ago, we're talking about how stress poison our body. So I just want to take this opportunity to ask you, I don't know much about that. Can we have stress as a poison to the body?
Stress cannot be exactly taken as a poison to body, because let's say, there are a lot of things which actually gives you stress. If you're having an extra workload, if you are going here and there, and then if the things are not going right in your life, a lot of things are there. But then you exactly saying that stress is gonna act as a poison to your body, it can be taken as a sort of poison, but then this falls more under your psychological perspective rather than a toxicological perspective for that sake.
Okay. And then, ma'am, you explain how we get tolerance by the body when there is maybe an infection whereby these bacteria they develop, resistance to the drugs and all that. What about a situation whereby there is no any infection, like it's a habitual thing, for example, consumption of these street drugs?
Let's say if the body, how do I say it? If the body is having some infection, the bacteria that is there that gets habituated to it, and then they develop tolerance. But then if a person is consuming some street drug, or let's say alcohol or something on a regular basis, so what happens? The body or the brain starts thinking that is what the body is supposed to take on daily basis, and that is how they start developing their tolerance. Then if the person becomes a chronic abuser for that thing, and then with a small dose which was actually giving him a high or a kick in starting stages, that will not be giving the very same feeling to him in the later stages when he's just taking, let's say, 30 ml of alcohol or 40 ml of alcohol for that say. Then to him to get that very same level of kick, he has to take some 60 ml of alcohol. But then apart from this, I would also like to say, when let's say a person is has become a very addicted to some drug of abuse, let's say cannabis or for that say. Now what exactly is going to happen here is, if the person has become a chronic abuser for it, now suddenly the person want to withdraw, because that thing is completely altered every mechanism that is normally going inside his body. Now the person cannot normally withdraw from it, because there is something known as withdrawal symptom also which come into play. Withdrawals, now what we exactly understand by this withdrawal symptom is, let's say a person was a chronic abuser, and now because he was having issues, and then he's taken to rehab, and then it has been asked of him to stop taking. He cannot actually stop taking it in just one go. It take a whole lot of things. And then you might have also heard, or you might have also seen people getting very restless when they do not get those substances or those things as such, because the brain has quite acquired, yes, this is my regular functioning, this is my regular working, I have to work it that way, I need to get that substance. And that is how it happens in all the cases for that say, like be it acute, be it chronic, for that say.
Thank you very much, ma'am. I really appreciate it.
Thank you.
Okay, so we now have another question again in the chat box.
Yeah, this question is quite, how do I put it? I myself have a doubt in this question, to be very honest. If poison kept for a long, will it become more deadly or its effect reduced? I don't think its effect is gonna get reduced in any way. But then for me to answer this, I'll for sure get back to you after doing some research on this, how exactly this works. Because me, when I myself was a student as such, I had this doubt which has not been cleared from so long. But now I'll try clearing it. But then I need some time. I'll get back to you for this question for sure.
Or maybe Dr. Jan would like to add up on that.
Yes, sir, for sure. Dr. Jain can add on this.
I'm asking, I don't know if he's available and
Hello.
he has something to say.
Yeah. I'm here. Actually, I'm the one of the first people who is doing both medical toxicology and forensic toxicology.
Oh, wow.
So we are seeing patients actually coming with poisoning or overdoses. They come with those cases. So we see many times that this agricultural poisons, many of them are old poisons, or they have been actually kept in one side of the one corner of the room, and all of a sudden, because of impulse, this patient takes this poison. So some of these patients, they survive in spite of taking very huge amounts. Like for example, there is, and that is because of either the drugs, that active principle of the poison has now is lost its potency. So for example, aluminum phosphide. Aluminum phosphide is a tablet which is available in India, used for as a fumigant. This tablet is kept in all the granaries, and once it comes in contact with moisture, it releases out phosphine gas. So when the person has consumed an old aluminum phosphide tablet, which has lost its potency, it has already released the phosphine gas, there is more chances of the person to survive.
Yeah.
At the same time, similarly, we have got phosphorus poisoning also. Like for example, a small child, a girl child who was sweeping the floor, she came across this food material which was kept to attract the mouse or the rat, and this food material was already laced with poison, that is rat poison. But no one could see from the just by viewing it, no one could understand it was already having poison in it. So this child innocently she just consumed this food particle which was lying on the floor, and that is a different scenario. She actually went into liver failure, but it was also we didn't have to go for aggressive management. So from these two cases, what I'm trying to say is that poison's potency, it will get lowered because it's a chemical. It reacts to various substances which it comes across, like which on the process of before entering our human body, if it's coming across a lot of other like diluents like water or some other substances like dust, so many things, and it's already reacting to even moisture. So its potency will definitely get reduced. That may be the reason why these patients survived in spite of not getting this aggressive management. Thank you.
Thank you so much, sir. It's actually great.
Thank you so much. So now we can now have the final closing remarks, Miss Richa.
So what I was saying is, I think we should be, very aware of what exactly is happening in our surrounding. What pollutants are we getting exposed to? It's not like we are all very safe, or we are, the environment is very safe. We are getting exposed to one or the other thing on our daily basis. Let's say we all are very adventurous. We all like to, go on hiking and stuff, and then while going hiking and stuff, we randomly see some plant and we just want to, touch because it's beautiful. We should never do it. You never know what poison is that plant holding and what it can lead to you. So always be very aware of what is happening in your surrounding. Never touch anything you just like because it's looking very beautiful or anything for that same. And then be very cautious of what you are taking, what quantity of it you are taking. Just do not come into your peer pressure and then just be like, yeah, we should take it, it's good. No, never do it. Think straight. There is always this instinct which come from inside, which will, lead to what exactly is right and wrong for you. And then please do listen to your instinct and be very aware of what is happening around you. That's all. Thank you.
That was great. So to close the session, first of all, I would like to thank our speaker for her wonderful session.
I hope so. I would have done justice to the topic.
Oh, you did very great. That was very impressive. So thank you so much, Miss Richa. Your lecture was very, very intriguing and also comprehensive, very, very comprehensive. We appreciate the efforts, the time and commitment you kept towards making this lecture an amazing one. We look forward to having you in the future to talk about things related to toxicology or even forensic science as a whole. We hope to have you in the future again because we really enjoyed this session. And I cannot complete this session without sending special thanks and acknowledgement to Dr. JAIN for his great contributions as well. Thank you so much, Doc. And lastly, to our amazing and wonderful audience, thank you so much for your time and patience. We are grateful for you staying with us throughout the session. We hope to see you again in our future session. That is our next session in January, right? The year is ending. So we are ending our year with Ms. Richa Singh on forensic toxicology. Hopefully next month, next year, we would be updating you on what we'll be doing next for our episode 8 on the forensic post lecture series. Thank you all. Have a beautiful and a great weekend, as well as a new year. Happy New Year in advance. So without much further ado, I would like to close the session. Thank you all once again.
Thank you. And thank you, audience. And thank you to JAIN sir for so much of insights.